I will try to explain to you now something very new. It is a selective peroxisome proliferator activated receptor alpha modulator, SPPARMA alpha concept. I would like to come back in the beginning of 90s. In my group with different bright scientists like Patrick Durier, Bart Stals, Johan Overks, my wife Jamila Najib, and many others, we demonstrated for the first time that peroxisome proliferator activated receptor regulate genes governing lipoprotein metabolism, but also vascular inflammation and atherosclerosis. This was confirmed by different groups, Walter Wally, and many thanks, Walter, to come from Singapore for this meeting, and also Peter Libby and Peter Libby group. And it was the beginning of a nice story. To make this story short, in fact, PPAR alpha is a nuclear receptor. It is mainly in the liver, but also in other tissue, and also in vascular cells. And after ligand binding, for example, by a fibrate, but also with a fatty acid derivative, there is transactivation. It's a DNA binding dependent activity with activation of the transcription of target genes with a target gene promoter, and transrepression. It's a protein-protein interaction, totally different from transactivation, with repression of the transcription of genes like NF-kappa B. It is not only for PPAR, there are many transcription factors, and they all act like that. So the first step, the ligand binds to the nuclear receptors, PPAR alpha. Your activation, you have also influence of RXR, and this activated PPAR alpha induce a three-dimensional conformation of the receptor that leads to selective recruitment of different cofactors. When we started in Pasteur Institute this work, we had only few cofactors. Now there are 328 cofactors. So it is more and more complicated. Transactivation, it was very nice because step by step we demonstrated that after binding of the zinc finger DNA binding domain to the PPAR in different genes, you have transactivation. And what was fascinating for us is that the protein which were increased by this transactivation were very important for lipoprotein metabolism and fatty acid metabolism. apoA-I and apoA-II, the main protein of HDL. apoA-5 and lipoprotein lipase, so important for triglyceride-rich metabolism. SRB1, ABCA1 for reverse cholesterol transport. CD36, FATP, SES, CPT1 for fatty acid metabolism. And also other transcription factors like ELIXA, reverb alpha with a linkage to ApoC3 modulation. So it was very fascinating for us to see that. So by increasing apoA-I and apoA-II, by increasing ABCA1, there was a profound effect of PPAR alpha agonist on HDL production. And by acting on ApoC3 and apoA-5 on triglycerides via free fatty acid, it was very interesting to look at decrease of VL production, increase VLDL clearance, decrease LDL particle number, increase LDL size. But if you want to summarize what does PPAR alpha activation, it improves dyslipidemia, and this is very important, but it decreases also very nicely vascular inflammation. And last but not the least, we demonstrated in our labs that PPAR alpha agonists regulate the expression of key genes involved in triglyceride metabolism, as Anne mentioned today, apoC3 gene, LPL gene, apoA-I gene. So now I would like to move to the rationale for SPPARMA. Why to develop a selective PPAR alpha modulator? In fact, if you look to the history of fibrate trials, the first trials were done with a very interesting compound, the gemfibrocin. These trials were positive, but unfortunately, there are safety issues when you combine with statin. And if you cannot combine with statin, you cannot use in diabetes, for example. So trials with other fibrate like fenofibrate were negative, except for the subpopulation with atherosclerosis dyslipidemia. And in addition, we knew since many years that fenofibrate and other fibrate increase creatinine and hepatic enzyme, and it is not very good. In fact, in 90, I was looking with my team to some interesting nuclear receptor, estrogen receptor. And at this time, a very fantastic chemist, Lewis, did a wonderful work to separate beneficial action of estrogen receptor agonist and negative action of estrogen receptor agonist. And it was a beginning of tamoxifen and particularly raloxifen, which is used now, still used in clinic. And in fact, it was interesting that you can do research to create ideal receptor modulator to improve quality of life or prevent diseases. And in fact, the paradigm on which the search on selective nuclear receptor modulator, because it is the same story with estrogen receptor and PPAR, is based on this cartoon. In fact, if you have different ligands, for example, if you have 100 different fibrates, if this fibrate bind to PPAR, but after binding, they induce different conformational transition. It means that the PPAR alpha is different in conformation. And this conformational transition induce a specific recruitment of co-activator, co-repressor complexes. And by this way, you can have a selective activation or repression of a subset of genes. So this is a normal concept, and the SPPARMA concept is the basis of SPPARMA alpha concept. So what is this concept? So this concept is it must be possible by selection of agonists to activate the transcription of desirable target gene and to activate the repression of undesirable genes, and by this way to increase efficacy and to increase safety. So the SPPARMA alpha concept is based on drug-modulated cofactor activity. The binding of different ligands to nuclear receptor induce different conformational changes. Consequently, the binding affinity with cofactors, and there are many, may differ. And each drug, in fact, has a characteristic cofactor binding pattern. If you look to all fibrate, no one is the same than another. So different cofactor recruitment may modulate different physiological pathway, tissue selective effect, gene selective effect, and the drug effect is correlated with a cofactor binding pattern. So in order to be more precise, for example, if you consider a ligand 1, for example, fenofibrate or bezafibrate, and a ligand 2, another fibrate, ciprofibrate, you have binding to PPAR alpha, you have shared cofactors, but you have also specific cofactors. And at the end, and you will see that with the lecture of Professor Kodama, you have shared biological response, but you have also distinct biological response. So the ideal SPPARMA alpha, we were in 2001 for treatment of CVD and atherosclerosis. In my mind was a selective agonist, so pure PPAR alpha agonist with very high potency and no side effect. It was a dream. You know, sometimes you wake up and you say, I have a dream. But this dream was possible because COA, a pharmaceutical company, I knew the president of COA, Mr. Miwa, and I exposed to Mr. Miwa my dream. And he said, I can put all my chemists with you. And they can generate 1,000 different PPAR alpha agonists. And perhaps, as you say, one of them will be a SPPARMA alpha. It was very risky. It was a gambling research. But we did it. And in fact, I asked Dr. Kantanabe in COA laboratory to do 1,000 different patentable PPAR alpha agonists. And they did 1,300. Can you imagine? The chemists in Japan are fantastic. And the problem was pharmacological profiling. And again, it was a huge work. In fact, with the team of pharmacology and with Dr. Abe and COA laboratory in Tokyo, we use that. We use, for each compound, we were looking by reported gene assay on PPAR binding properties. We look at activation of the transcription of genes in target cell and tissue, gene profiling, and microarray. We look to undesirable side effect on glucose, lipid metabolism, vascular effect, anti-inflammatory effect, by in vitro and in vivo on transgenic or knockout animals. So, and we were looking to cofactor recruitment properties by GST, pull down and fret assay. A huge work, 2 years. But at the end, it was a miracle. The key 877, and not the other one, was a SPPARMA alpha agonist. In order to demonstrate that, I would like to express my gratitude to Japan atherosclerosis societies and to all professors in Japan in clinic. They did a fantastic work for phase 1, phase 2, phase 3, and you will see in this meeting what they did and how they demonstrated that this compound was so interesting. We had a very good experiment in animals, because in my lab with Professor Smith's Nobel Prize and Maeda, we developed apoA-I to knock-in mice fed with a Western diet. It is a wonderful model of dyslipidemia. And when you give K877 to these mice, you have a splendid effect on cholesterol, triglycerides, HDL. But in addition, you have very good effect on atherosclerosis development. You suppress atherosclerosis with Pemafibrate when you compare with fenofibrate. In addition, many thanks also to Professor Yamashita. He did many work in human, and K877 compared to fenofibrate was better to increase not only HDL, but small HDL and cholesterol efflux in human primary macrophages and reverse cholesterol transport in human apoA-I transgenic mice. And you will see other experiments done by Professor Yamashita in Japan on this topic. Now, the problem is we had a very good compound, but what is the potential for clinical use? In fact, to understand the originality of this compound, I would like to come back to the structural organization of nuclear receptors. In a nuclear receptor like PPAR-alpha, you have DNA binding domain, DBD. You have ligand binding domain, LBD. And we know everything on the conformation of this ligand binding domain. So when you consider the DBD, DNA binding domain, with this gene with HGG-TCA, which this is the signal for the binding. And when you consider the ligand binding domain with receptor, PPAR-alpha, and retinoase X receptor, you have transcription. Look at that. This is beautiful. The specialist of conformation did that. And you have in green PPAR-alpha with binding to DNA. In blue, the RXR-alpha. And in red, you have the cofactor. And the binding of the cofactor are dependent of the structure of the LBD. And what is fascinating for me is that after the binding of the ligand, you have a displacement of the H12 helix. And this movement stabilizes the helix and stabilizes the LBD. As I said before, we know now 320 cofactors. It's not the end of research. You have the young people in the room have many things to do. And 38 were identified to bind to PPAR-alpha. But among these receptors, one is very, very important. It is PGC1 alpha, because dysregulated PGC1 activity predicts systemic insulin resistance, glucose intolerance, and insulin deficiency. This is the transactivation. You have here the molecular explanation of gene transcription, and you have the role of PGC1. But what is very interesting is there are potential contributions of organ-specific dysregulation of PGC1 alpha to the development of insulin resistance and type 2 diabetes. And in PGC1 knockout mice, you have a high influence on insulin sensitivity and insulin resistance in different tissues. So I would like now to come back on the structure of different PPAR-alpha agonists that we studied, like fenofibrate, and that we developed, like K877 with COA, but GFT505 with GenFit. My wife, and when she was director of research in GenFit, develop this compound. It is also a little bit SPPARM, but it is not a pure SPPARM alpha because there is some delta. And if you look to the EC50, you see that K877 is 10,000 more potent than fenofibrate in vitro. When you use fenofibrate in clinic, you use 200 milligram. When you use K877, you use 0.2 milligram. It's totally different. And many thanks to the wonderful work of Mr. Yamamoto in the group of Professor Shimano, who is here, because they elucidated, beginning of this year, the molecular explanation of the activity of K877. In summary, when you have fenofibric acid coming to the LBD, it's very large. It's very large binding. Not too many amino acids are involved. And it is a tight linkage. And the cavity size after binding is 1,163 angstrom. It's not good. But when you look to K877, you have so many binding because that, the conformation. You have so many binding, and the cavity size is 828. So you have a fantastic change in the binding domain. And this is the explanation of the effect. Because compared with the classic fibrate, K877 bindings more efficiently stabilize LVP, stimulate stronger interaction, and interface with PGC1 coactivators. And the valine 306 play a major role in this interaction, this valine. Now, I don't want to present too many data, but in 2 slides, I would like to mention why K877 is so interesting. If you look to existing PPAR alpha agonists like fenofibrate, bezafibrate, ciprofibrate, you have a decrease of triglycerides. You have an increase of HDL, but not too much. You have a decrease of apoB48 and RLP, but not too much. With the SPPARMA alpha, you have more and more decrease of triglycerides, more and more decrease in remnants. You see, 55% for apoB48, and you have a large increase of HDL cholesterol. But what is more interesting, particularly with the development of NAFLD and NASH, is that if you look to the side effect, with classic fibrate, you have increase of hepatic enzyme in many patients. With the SPPARMA alpha, you have not only increase, but decrease in ALT and in gamma GTP. And if you look to creatinine, it was a huge problem for FDA when they wanted to recognize fenofibrate. There is no increase of creatinine with the SPPARMA alpha compound. But with Professor Aikawa, thank you for him, is leading the COA research lab in Harvard with Peter Libby. They demonstrated they have preclinical evidence for the effect of K877 on vascular diseases. And he will present to you fascinating results on different animal models and cell models. So a new generation of potent selective PPAR agonists have the potential to improve the management of dyslipidemia, CVD, and to prevent the development progression of diabetic vascular complications. Thank you for your attention.